Okay, so we are going to move on. And up next, we have Dr. Brianna Gaskill. Hang on a minute here. So our second speaker for this session, Dr. Gaskill, if you want to show your slides. Dr. Gaskill, if you would like to show your slides.
Yep, I'm on it.
All right.
There we go.
Okay, here we go. So our second speaker today is Dr. Brianna Gaskill. Her research program at Purdue University focuses on welfare assessment of laboratory animals. She utilizes natural behavior, physiology, and affective state to assess an animal's overall well-being. She is especially interested in how better welfare can translate into better and more robust science. Her research interests include applied ethology, enrichment design and application, improving husbandry techniques, and how environment can affect scientific results when not tailored to the animal's needs and motivations. Her presentation today is on the state of the art in rodent handling. Dr. Gaskill.
Thank you, Pat. Can you hear me okay? Can you see the slides? They look good?
Yes.
Yep.
Yes.
Okay, great. Well, thank you all for hanging in there till the very end of the day today. Hopefully I'll wake you up a little bit with some good videos, and I have a few surveys that hopefully will mix things up a little bit so that hopefully you're not snoozing at your desk. But then again, I don't know, so have a good nap. Anyways, so I'm going to be talking about some, in a very broad overview of some of kind of the latest techniques with rodent handling. I'm going to talk about some of the things that Brianna was just speaking about, and hopefully give you a little bit of idea of what are some techniques that are out there that people are using. I'm going to sprinkle in a little bit of data just because with the short timeline. So if you have questions, definitely make sure to ask. So I wanted to start off here with this image because this is Dr. Hans Selye, and he is affectionately known as the father of stress physiology. So he discovered all of the hormones that Brianna was just talking about, corticosterone, cortisol, and the entire HPA pathway. Now, he discovered this because he is a horrible animal technician. And it really illustrates the importance of animal handling. So for instance, he was studying the effects of various extracts, and at that point looking at various hormonal impacts on animal physiology and behavior. And so he was testing this extract on some rats and was injecting them on a daily basis, and really wasn't great at animal handling. Sometimes, at least as the story goes, he would occasionally drop the rats and have to chase them around the room in order to pick them back up and get them injected. So in essence, he was a really bad animal handler. Well, the outcome of these studies was that he found enlarged adrenal glands, thymus, spleen, lymph nodes, and he thought, oh my goodness, I have found this new hormonal extract that is affecting the body, and I'm really excited. Well, the good thing is that Dr. Selye was a really good scientist. He may be a bad animal handler, but he's a really good scientist. And so he compared these results to some other extracts, and yet he found the exact same results. And so luckily he looked into this and ultimately found that it was the way he was handling the rats that was affecting these physiological responses and not necessarily the thing he was injecting them with. And so this led to his entire career understanding stress physiology. And so I think it's a really good emphasis for us to understand that the reason we know about stress physiology is because of the way he handled rats. And so this is a really, really important for the work that we're doing. So it's obvious that rodents, especially when they are naive to human handling, they're stressed. And they're afraid of us because they don't understand. It's like potentially being handled by a T-Rex. You don't know if he's going to eat you at any point in time. And so obviously the things that we need to do to the animals in order to further our biomedical research can maybe exacerbate these fear responses. And obviously this fear can lead to changes in physiology and overall animal biology. That's obviously going to affect the welfare of the animals as well as the scientific quality that we're extracting from these animals. I probably don't need to go into this, but there's so many measures that can be easily affected by the stress response. I've got some data here that's showing some animals that have been injected and actually via, I believe it was a catheter. So they weren't handled. So they were injected with a lipopolysaccharide that creates this cytokine storm and essentially a fever response. And we see this beautiful triphasic fever response that actually for the longest time before this study was really not believed to exist in rodents, that they had a different fever response than we would expect in humans. And when you compare that to these animals that were handled, they were injected the same way, but they did a faux injection of the LPS. You can see that the overall shape of this fever response is quite significantly different, and you don't see that really nice triphasic data that you do in the first graph. And even looking at the black line, so the animals that were injected with essentially, a control, a vehicle, you can see that just the fact of handling, you see a spike in their core temperature just by being handled. So this can have a big response on the data that we're collecting. Even as far as looking at gene activation. So this is brain slices from the paraventricular nucleus and the hypothalamus looking at CFOS activation. So in these two animals, the difference is that one of these animals was on the left, the naive, in box A. They were not handled and immediately, so they were taken immediately to were euthanized, and the samples were taken, whereas the animals that received the acute stress were scruffed, left for 2 hours, and then were euthanized in order to get these brain slices. And you can see the acute difference that even something as simple as scruffing can have on whole animal biology. So there are a lot of procedures out there that people use in order to try to minimize this effect of handling stress. So you might call it habituation, some people call it gentling, and it's common practice, especially more often in the behavior realm, to utilize a lot of these techniques because handling has such an immediate and robust effect on the animal's responses in a lot of these behavioral tests. It's very, very sensitive to stress. And so Maurer et al., the images that I'm showing here, found that touching, stroking, lifting, talking, and hand feeding the rats various types of treats during this gentling procedure, spending up to 4.5 hours per cage, reduced overall fear responses, improved the ease of handling, and they saw these responses up to 6 months, which was great. However, using this particular protocol to habituate just even 10 rats would require up to 40 hours of dedicated employee time. And obviously this is going to be a challenge to really get large scale implementation. But it is something that is time intensive, potentially costly, but it is effective. So this is something that a lot of people have been utilizing, but there are some new techniques that have recently come out in the last year and a half by this fantastic group out in Sweden. Unfortunately, there's no necessarily published data with this group, but they have some amazing videos of animals really almost volunteering to participate in procedures. And so in this video that I'm going to show you, is a woman who is ready to give some rats an oral gavage. And I want you to watch the behavior of the rat that gets selected, and I want you to watch the behavior of the other rats actually in the video. So we'll see if we can get this going. So this animal is pretty calm on this little mat, doesn't have to be fully restrained, appears to be pretty calm during the gavage, and we're done. Now, the thing I love about this is the very end, those rats that are getting ready to be oral gavaged are almost desperately trying to leap onto that fuzzy pad. And actually, it's this fuzzy pad that's the key in this particular video. So what this group is doing is they're using something that's called positive conditioning, place conditioning. And what they're doing is they're taking this fuzzy mat and they are giving these animals all kinds of positive experiences, lots of tasty treats, they get to forage in it. And these are the experiences that they associate with this location. And it's actually something that's very basic learning theory, animal learning theory across the board. And so they're using this in order to, when they need to do these procedures, the animals immediately think something positive is going to happen. And even though, as you saw in that rat, he didn't seem to be too distressed by having this thing done to him that maybe had never been done before. I'm not sure how many times that had been done with these rats, but as you can see, even if it has been done multiple times, they don't seem to be distressed about being manipulated by their caretakers. And they have a bunch of really, really great videos on the NC3R's International Animal Technicians Symposium that they held last year of some of these different techniques that, not just with rats, that they're actually doing with mice. And actually, there's a video here looking at a mouse that doesn't have to be restrained at all for a tail vein blood sampling. And really, in general, the animal seems to be fairly calm. Obviously, it reacts a little bit to the stick, but in general, this is pretty good. So they've got some really lovely videos and information on this webpage that I'd really recommend to check out, because there's some really cool ideas in there. I know they've got a handout that they are sending to people, and if somebody's interested, I think I've got it somewhere, about general procedures, but essentially it takes a few weeks for them to essentially train the rats and the mice to condition to this particular fuzzy mat. So it's a really cool example of what some people are utilizing right now. So that brings us to our first survey. And essentially what I'm wanting to ask is, does anyone actually use any kind of conditioning, positive reinforcement training with their rodents specifically? And so Camilla, if you can bring up that survey, that would be great.
The responses have stopped, so I'm going to end the polling and share the results.
Okay. Wow, that's actually more than I expected. I've not heard this utilized very much with rodents altogether. So the fact that 52% of everybody on this webinar are sometimes using it is actually better than I expected. So that's pretty cool. I've utilized it myself for various injection techniques where the animals were given a positive reward, chocolate milk, which made some very entertaining videos. So it's cool to see that some people are trying this. So that's exciting. So let's move on to our mice. Oh, make sure we're moving here. Okay, there we go. So let's talk a little bit about mouse-specific handling techniques that are available. And this flows very well off of the techniques that were just being spoken of. However, because I knew she was going to be talking about cup handling, I'm not really going to talk too much about the cupping procedures that Dr. Jane Hurst and her lab developed. So I decided to go ahead and focus on the tube handling, which seems to be more of the push from Dr. Hurst as well as NC3Rs as the methodology they seem to really want people to start utilizing. And so this particular handling method looks something like this. The animal is basically shooed into this clear plexiglass tube and can easily be moved from cage to cage, can easily be tipped onto a cage lid in order to do a scruffing procedure. And very similar data that were just shown. This really has a big impact on the behavior of the animal. And so in the work that was done by Dr. Hurst, we see that animals that were tail handled, so the traditional base of the tail handling compared to the tunnel handling, that we see that these animals are more likely to attempt to interact with a human hand. So they did a basically a hand test where a hand was placed within the cage to look at voluntary interactions or percentage of time the animals seemed to be interacting with the hand. And so that was significantly increased with the tunnel handling, and also had been increased with the cup handling as well. But we see that it also reduces fear behaviors, very similar to what we were just seeing with the cup handling method. So looking at stretch to 10 postures within the elevated plus maze. This is data from their BALB-Cs that they tested, but they tested several different strains. And we see that overall, we see a reduction in fear-related behaviors due to this particular technique. And they've since published several more papers looking at how it affects various aspects of physiology and understanding how these can be implemented. And in fact, again, the NC3Rs has really tried to adopt this particular type of handling methodology and have a series of sites that are dedicated to really improving low stress handling. And so they've got some videos, various tutorials, they've got some posters that you can actually request that you could hang in your vivarium. And some really nice resources that if you're trying to convince a PI to potentially think about utilizing this technique, because as we just heard, it might affect some of their physiological metabolic markers, they have published literature that you can grab in order to share with these researchers. So this is a really nice location to get some of these resources, to find out more information, to get a little bit of training and see how maybe you could implement that within your vivarium. So that leads us to our second poll. So Camilla, if you could please pull that up. And it has to do specifically with tube or cup handling. So are you utilizing this currently in your vivarium? Again, higher than I was expecting. This is really encouraging to see that these are starting to become implemented more readily within the United States. I think even over in the UK, they're a little frustrated with the fact that with all of this really great data showing that these techniques have impact on the animal's behavior, on physiology, on their overall welfare, that they're not necessarily being implemented necessarily as quickly as they had hoped. So that's really encouraging. I'm excited to see that you're at least using them sometimes. So next, we'll move on to looking at some techniques that are specific to rats. And I'm probably going to spend just a little bit more time here than I did on these other techniques, simply because this is something that my lab has really been involved with in the last few years, and looking at the technique of rat tickling. And hopefully some of you have heard of it. If not, tuck in, we're going to get to some fun videos. So rat tickling was developed by the late Dr. Jaak Panskep, and he was considered the father of effective neuroscience. He was really interested in developing techniques to systematically study the neurological basis of positive affective states, or essentially emotions. And really they were interested in studying the foundations of human and animal emotions, and specifically positive emotions. And so they were hoping that identifying and understanding these positive emotions might confer some resilience to depression, anxiety, as well as lead to some overall increases in overall general health. And so he started looking specifically at one particular behavior, which is play. And hopefully you can see this video. Young juvenile rats will spend a large amount of time engaging in social play. They're an extremely social species, and this behavior is very dynamic. Actually, you can see even up into adulthood that they will engage in this play behavior. And so the technique of rat tickling really is mimicking this two main aspects of rat play. And so really we're not tickling them, we're just mimicking rat play. It just has to do with part of the technique that tickling came from. So some people prefer, we call it heterospecific play because it sounds science-y, but it's the same thing. So actually what we're doing is we're mimicking these two aspects, the dorsal contact, which is what you see on the top, and then the pin on the bottom. But we're mimicking this with our hands. So using our fingers, so this is where the name came from, you're using your fingertips in order to make that dorsal contact on the nape of the neck. And then we pin the rats, which freak people out a little bit because they think we're being really rough and are we sure that they like it, but it's actually called rough and tumble play for a reason, because it's very dynamic, it's very energetic, and so really, and what we found is that the pin is maybe the most important part of the technique that the animals really respond to. And so we know that they like it because they give off these 50 kilohertz vocalizations, which are reflective of positive affect. And this is the thing that makes rats so, so cool, is that we have these indicators of emotional valence in rats that have been very well studied and validated that we really don't have in any other species. And so what we look for are these very specific vocalizations. So looking at a sonogram, there are these frequency modulated calls, which are called trills, and they sound something like this. So it sounds almost kind of like bird chirping. And so they give off these calls when they're eating tasty treats, self-administering euphorogenic drugs, and when they play. And so Dr. Panskep spent a lot of time studying these calls and the neurology of them, and found that these are extremely predictive of dopaminergic activity in similar areas of the brain as reward centers in human brain. And so he was able to show that not only are they predictive of activation of these areas of the brain, but they're actually correlated with reward value. So for instance, if I offered a rat a particular a peanut, he was completely full. He'd already eaten a whole meal. We wouldn't get as many 50 kilohertz calls when I gave him that peanut when he was full versus if he was hungry. And so we can see that the reward value changes and the number of these vocalizations changes based on the overall affective state of the animal and the reward of the experience. And so rat tickling looks a little something like this. So this is Franny. She's from San Francisco, and she is maybe one of the best rats we've ever tickled. And I want you to pay attention to the number of calls that she's giving off during the technique, and you can get a little idea of what this looks like. So Franny really likes being tickled, and she was really, really fun to tickle. And so, yeah, it may be something fun to do, but is it really something that's beneficial in the laboratory? So we have found that it is a very effective intervention. So my fantastic graduate PhD student, now former PhD student, Megan LaFollette, did a systematic review looking at the literature to see what kind of outcomes we were finding from the different projects that were being done with rat tickling. And what we found was that overwhelmingly, almost all of the studies found a positive effect from tickling, specifically measuring these 50 kilohertz vocalizations that are really indicative of positive affect. We see that the rats are more likely to approach a human hand if they've been tickled. We also see a reduction in fear behaviors as well as ease of handling. And so this really seems to be an effective method for improving animal welfare, reducing stress-related experiences. And I've got a video to show you just that. So a colleague of mine, Dr. Sylvie Cloutier, did a series of projects looking at whether tickling before an injection would affect the rat's behavior, measured vocalizations. And so I want you to watch the behavior of this rat as it's been injected. So as you can see, he's not really cowering this corner of the cage. He's just previously been tickled, gets restrained, doesn't seem to really be struggling. Even with the injection, we don't see any struggling. Replaced back in the cage. Now watch how close he comes to the hand and immediately wants to go back to playing and being tickled. And so you can see that this animal was most likely not under any amount of stress due to this procedure. So this is a really cool opportunity for us to reduce stress, but actually improve the ease of us doing these procedures. Unfortunately, even though this technique is really, really beneficial, we found that 55% of personnel that were surveyed in a survey that we put out, I believe, in 2018, at the beginning of 2018, we found that 55% of respondents in our survey of over 800 people never utilized this technique, even though it's been around since about, the first publication was maybe about the year 2000, 1999. And so it's been around for 20 years, and the majority of personnel that were surveyed are not utilizing it. So this was a little distressing for us. So we wanted to know, okay, so if 55% are not using it, well, why aren't they using it? So we asked essentially, what are some issues that you see with implementing this technique and identifying what those barriers might be. And we found that 59% of the respondents in our survey said that time was one of the major barriers to implementing this, which is not surprising. That's something that we hear all the time with implementing techniques or new enrichments, things like that. That wasn't surprising. But it was nice to have that data to show that. And so interestingly, the second most common one was what we labeled as personnel, but really what it was looking at various comments about perceived value of the technique by others, whether the personnel had adequate amount of training in order to do this procedure or knowledge about it, and as well as just overall education on the procedure. Why was it important? What were the benefits? And how do you do it? And then the last had to do with research-related parameters that are probably not things that we can adequately address, because a lot of comments were like, well, I don't want to utilize this technique because my animal has surgical implants, which makes sense. It's a very, you know, exciting, energetic behavior that there could be problems with engaging with an animal that might have an implant or something like that. So we started asking questions about, well, can we address these barriers in order to improve the implementation of this technique that's really so effective? And so one of our first questions had to do with that time aspect, that time barrier. And so based on Dr. Panskep's published SOP, we couldn't find any data that showed that this was something that they had studied to figure out how much time and input you needed to make in order to get those behavioral outcomes. And so we decided to do a dosage study, essentially with tickling, and see how much of time per session do you need to put in. So the duration of tickling, so do you need to do it for 15, 30, or 60 seconds in a session, and the number of time, the number of sessions that you do. Can we fit that within a traditional 5-day work week? And this was actually the maximum SOP that Dr. Panskep had. He said that you need to tickle for 60 seconds for 5 days. And so we were wanting to see, is there an opportunity for us to get the same outcomes at a smaller time investment? And what we found was that there was no effect of duration. So it didn't matter if you tickled for 15, 30, or 60 seconds, but it had to do with the number of sessions. And we found that you see a significant increase in the overall measures that we did. So we measured the 50 kilohertz calls, as well as looked at behavior, both in anticipation as well as after an injection. And we found that the most efficient and effective rat tickling investment was 15 seconds for 3 days per rat. And so this was pretty exciting, as that was an over 1000% reduction in the amount of time investment. So we were really excited about that. We can get away with this easily within an acclimation period when animals come into the lab and can't start on the study. Something that it is still a time investment, but this is faster than that 40 hours that we were talking about with that gentling procedure. 15 seconds or 45 seconds in comparison to 4.5 hours per individual animal. So that's a pretty impressive and a very efficient technique. So we next moved on, and we're kind of thinking about addressing that last barrier of knowledge, of training. And so Megan, my graduate student, developed this interactive rat tickling certification course as part of one of her studies, where we wanted to determine if providing information to people about the technique, the background, the science, and how to do it, if that would improve overall implementation in an actual human-based study. And so if you're interested, that's actually the website. It's freely available for you to take. And so you too could impress your friends and colleagues with a rat tickling certificate that you can proudly hang in your office. So if you're interested, you actually get a certificate of completion at the end of the course. But in terms of the data that we found, so we ran this study where we had 3 groups of people, some people that were interested in learning about that technique, and they were put onto a wait list. We had a group of people that only received the online training, so that certificate course. And then we had a 3rd group that got the certificate course, but then also were supplemented with a in-person hands-on training that was specifically about the technique itself. So how is it done, giving critiques, things like that. And so we see with this first group at baseline, there's really no difference in overall implementation between our 3 groups. But after they received their training, so either the certification course or after receiving the certification and their hands-on training, the only group we see a significant increase from baseline in implementation is our hands-on group. But then we also decided that we wanted to do a follow-up 2 months later, because it takes time to figure out how you're going to implement this, make sure that you get the proper approvals, as well as making sure that you have an opportunity to do it. And so we did a 2-month follow-up after their training and found that actually we saw an improvement in, significant improvement in both our online and our hands-on groups. And it's interesting, we saw a slight increase. It's not statistically significant from their baseline values, but in our waitlist group. And we think that that has something to do with, obviously we have resources in my lab available online. Maybe they looked it up. Maybe they did their own homework in order to try to implement this particular procedure. But we were really excited to show that by providing this information and techniques, that even with just an online course, we can get improvement in long-term, relatively long-term implementation of the technique. So we were really excited about that. So of course, I want to know if anybody else is using this technique. So this is our last survey. So how often do you tickle your rats? This is actually worse than the other procedures. I find that interesting. I'm excited about the 5% of you that say always. That warms my heart. So actually, we're not actually expecting to see it go to always, because our recommendation usually and in the certification course is that you put in those 3 days of time investment, and then once a week at cage change, you supplement the animals with a little bit of tickling as you're placing them from one cage to another. And so we don't ever think that we'll get to always, but the sometimes is actually what we're shooting for, because we don't really expect people to use it all the time. But good to know. So with that, I'm actually done with my talk. So again, thank you for the organizers for inviting me to talk a little bit about some of these techniques, my amazing grad student, Megan, who did all of the rat tickling work, and then the funders for that work as well. So I'm happy to take any questions during the question and answer session.
Awesome. Thank you very much, Brianna and Paranaz.